Well, it's my pleasure to introduce Brittany Adamson from Princeton. Dr. Adamson is an assistant professor in the Department of Molecular Biology and the Lewis Sigler Institute for Integrative Genomics at Princeton University, and a member of the Genomic Instability and Cancer Genetic programs. Dr. Adamson started her training at MIT in the laboratory of Angelica Amon. She earned her PhD from Harvard Medical School, where she joined the lab of Steve Elledge, where she used functional genomics to study DNA repair in human cells. And after graduating, Dr. Adamson joined the lab of Jonathan Wiseman over at UCSF, where she was Damon Runyon Cancer Research Foundation postdoc fellow. And her postdoctoral work really pioneered approaches for functional genomics. And I know that it was a huge source of inspiration for a lot of the work that goes on in my lab today. Now at Princeton, Dr. Adamson's laboratory continues to use and develop cutting-edge experimental tools, including genetic screening methods and single-cell RNA sequencing to study genome editing and DNA repair. And she is also the recipient of the Sear Scholars Award and Rutgers Cancer Institute of New Jersey New Investigator Award. So we're really happy and honored to have Dr. Adamson here today.
For the invitation and for the kind introduction, and to everyone for taking time out of their day to come hear about what we do. Where to start? Okay, so since we're a small group, please just stop and interrupt me with questions. I think it's more fun that way, and that way if I'm running late at the end, I don't feel bad. So, okay, so I'm going to tell you today about some of our work on trying to understand genome editing technologies and what happens when we put them into cells. So let's start with like a super, let me make the slides go forward. There we go. Super broad overview. So this is not news to anyone, but of course, over the last 10 years, applications of genome editing have accelerated at a rapid pace, and in part, large part, this is due to the programmable nature of Cas9 and our ability to target to places in the genome. And so if we're looking sort of broadly at the impact that genome editing has had and is having, we see more and more applications in biotech that touch on issues of environmental sustainability. We're seeing it a lot in food production and agricultural sciences, biomaterials that can be produced from genetically modified organisms. Where my lab is really focused more and more on is in the use of these tools for biological research and the development of new technologies to ask new questions across going from animal modeling all the way across to studying genomics and genomic variation. And of course, if you've been following the sort of CRISPR-related news lately, we're seeing more of this, more and more in sort of the medicine space as well, including the first CRISPR-based approved genetic medicine, which the FDA approved in December of this year. And so these very broad reaching applications. And one of the things our lab is really interested is in understanding the fundamental mechanisms through which these genome editing tools work inside cells. And so in the group, we sort of talk about ourselves as cell biologists of genome editing, right? We care a lot less about how they work in a test tube, but what are the mechanisms that these tools are interacting with in order to do their job in the context in which we want to use them. And so to give you a sort of very, very broad overview of one example where sort of the ability of our tools influences in the genome editing space, influences the way we use them, we can point to, as I mentioned, this recently approved CRISPR-based therapeutic, which was approved in December for treating sickle cell disease. And then there shortly after was also approved for beta thalassemias. And so again, a very brief overview, but I think this gives a good idea of how the sort of limitations of our tools and how they work influence what we can do with them. And so sickle cell is a disease, it's a monogenic disorder. It was described, I think in 1949 as the first disease with a genetic basis. And sickle cell has this very clear mutation in the beta globin gene, which causes the red blood cells to sickle and causes all sorts of pathological effects. And so the way this therapy works, in very brief overview terms, is that an affected patient will have their hematopoietic progenitor cells taken out, edited in some manner, which alleviates the effects of this mutation. And effectively, it's a self-bone marrow transplant put back. And then the follow-up studies look really good as far as these cells behaving and strong mitigation of the disease effects. So what the CRISPR-based part of this therapy is the CRISPR is actually targeting an erythroid enhancer in this BCL11A gene. I think a lot of times when we think about genetic medicines, we think about a causative mutation and correction of that mutation back to something that would be non-disease causing. But in this case, actually, the work of Dan Bauer and Stu Orkin and lots of others in the field of understanding the gene regulatory mechanisms of the beta globin locus. The mutation is somewhere in the beta globin gene, but it turns out that if you mess up this erythroid enhancer in this transcriptional regulator, the stem cells will actually make this switch, and instead of expressing the disease beta globin, will express this gamma globin, which can rescue essentially a lot of the effects of this. Okay, so one of the reasons that this is such a great target is because of what our tools, the first generation of CRISPR tools, does, right? So CRISPR makes these double-strand breaks and causes gene disruption. It's really easy to target it someplace in the genome and have all sorts of insertions and mutations sort of stochastically made at that site. And so actually blowing up an enhancer is far easier than going in and taking it A and in a large percentage of cells making it a T, right? So in many ways, this is an ideal target, but one of the reasons why this was a great target is because we had this beautiful tool that was able to do this thing, right? And so this gives you this idea of how the tools that we have available to us can shape actually how we use them. All right, so that's the general introduction. But I think if we back up and think of an ideal situation, one of the things we'd love to do is take a population of cells, right, that has, and actually change one base pair in the genome of those cells in 100% of the cells from one thing to something else. Okay, and this might be the desired outcome of a lot of genome editing experiments, but what we actually end up with the vast majority of times is something that looks more like this, where you may want to take a T and make it an A, and you can do that with high precision and specificity in some percentage of cells, but in a lot of the other cells, you're either going to end up with an unwanted edit at that target or some sort of bystander mutation. And then in other cells, if your technology is not super efficient, you'll end up with unchanged sequence. And while this might be a feature in some applications, it would be great to have full control over this, this landscape in order to sort of dial it up or dial it down. And for those of you familiar with this space, this sort of paradigm is true no matter what genome editing technology you're using. In the vast majority of cases, this is true, as I just described, for programmable nucleases, right? So this is classic Cas9, where you can target it to a site in the genome, make a double-strand break, and the cell's own DNA repair machinery will stick a heterogeneous set of mutations into that site. The same is true for base editors. Some base editors can get really good at putting in the intended target, but they have bystander mutations and are not always 100% efficient. But what I'm going to tell you about today is our work on one of the newest genome editing technologies, prime editing, which was published roughly 4 or 5 years ago now, initially out of David Liu's group. And the thing that's unique about this technology is it makes intended edits what we want to make. So for example, that A to T with very high specificity and precision, although for the last several years, the field has been really struggling with this idea that it's incredibly low efficiency, okay? And so as a general philosophy in the lab, we think that by understanding how the individual edits are made and what the mechanisms are that influence these edits, we can then back up and think about engineering tools that produce more or less of the outcomes that we want in a way that we have more control over. And so as I mentioned today, so we've worked in the programmable nuclease and the base editor space, but today I'm going to focus on a recent story out of the lab, which hopefully will be online in a couple weeks, describing our efforts to improve the efficiency of these prime editors. Okay, so What I mentioned was really remarkable and sort of paradigm changing about the prime editors. And this is now true. Prime editors have been expanded into its own sort of class of genome editors. But what was really remarkable at the time when this was first published is that if we look at double-strand break induced editing, we can provide a template with which the cell at a double-strand break will incorporate an intended edit that we program on that template. And we can measure the rates at which the cell does that here in blue. And you can see those rates are generally pretty low when we're just doing this in cell culture, even when we're just doing this in cell culture. But there's always this gray bar of just insertions and deletions that accumulate at that site. Okay, so you have all these unwanted things, and then you have these wanted edits. In the prime editing space, Andrew Anzalone, who built this in David Blue's lab, really nicely showed in the first publication that rates are generally pretty low, except when we're talking about this one very special cell line 293Ts of the intended edit. But the remarkable thing is that the unwanted edits were very, very low, and in many cases hard to measure above background. And so instead of having a case in which the thing you want is produced in a background of a whole bunch of stuff you don't want, you have the case that the edit you want is produced in a background of otherwise untouched cells, right? Which to my mind at the time was this is a much easier, if what we're trying to do is do 100% precise editing, this is a much easier problem to solve than this for a variety of reasons. Okay, so how does prime editing work? So prime editing is essentially a Cas9 nickase hooked up to a reverse transcriptase. And so these guys are fused, although several groups in the intervening years have shown that they don't actually have to be fused. And what happens is a Cas9 hooks up to a guide RNA, goes to the target site, nicks it, just like a regular Cas9 nickase would. The guide RNA in this case is actually extended on the 3' end in such a way that it provides a reverse transcription template that this reverse that can actually prime to the released flap. And then the reverse transcriptase can transcribe whatever sequence we provide right next to the priming site into that strand on the genome. Now, if the regions surrounding an edit that you might want to incorporate into the genome are homologous to the genome itself, at some frequency, what'll happen is you'll actually incorporate that edit into the genome. And one of the reasons this is so precise is because we're not making double-strand breaks here, and it's a very directed process. As a matter of bookkeeping, so that you understand what's on the slides, the nomenclature in this field has gotten a little wild, and by the end of the talk, I'll make it worse. So we're going to try to just introduce it as we go. What I just described is called a PE2, the PE2 system. There's also a version of prime editing called PE3, where what happens is you put a nick on the non-transcribed strand, and this has been shown to make prime editing more efficient, just the presence of this nick. So the first question that my group wanted to ask in this context is, when we look at this, because we come from a background of DNA repair, what we see is that this is damage to the genome. It's not a double-strand break, but this is something that the cell is going to have to deal with and is probably going to involve the activities of DNA repair factors. And so the first question we wanted to ask is, what are the DNA repair factors and pathways involved in this process? And so we had also built out in a couple years prior to the publication of prime editing, a platform for asking exactly that question. What are the DNA repair systems that are involved in different genome editing processes? And the way this platform works, we call it RepairSeq. It's a CRISPR-I screening platform where you have a set of K562 cells that are preloaded with CRISPR-I machinery. CRISPR-I is a technology for repressing gene expression, and we can do this in large libraries for loss of function screens, where we deliver a guide RNA and a different guide RNA to each cell, and then the CRISPR-I machinery will turn off the targeted expression of the targeted gene to get a loss of function. And so we can build these large libraries where what we're doing is we're getting knockdown loss of function perturbations into each cell for DNA repair genes. And then right downstream of that guide RNA expression cassette, we put a cassette where we're going to target our DNA damage, or alternatively, where we're going to target our genome edit. We put these into cells, we select them up, and then we actually introduce our programmable editor, which targets that site right there. Once we allow the cells time to repair, we can pull that little piece of DNA out that has two pieces of information. One, the guide RNA for CRISPR-I, which tells us what the loss of function perturbation was in the cell that we're looking at. and then the target of the genome editor, which tells us what edit was actually installed when we did our genome editing, right? And so from that, we can build out a data set where we actually can look at the effect of every genetic perturbation, prescribed perturbation in our library, on every single possible repair outcome that happens at that breakpoint. And this technology has been really informative across a huge number of genome editing tools. The thing about Prime is I told you that predominantly we're making one thing, right? So when we do these experiments with Prime, we're really looking primarily at the intended edit and what modulates that editing event. And so in this case, what we were targeting is making a G into a C base pair at this particular site. Okay. So when we look across all of our genetic perturbations, what we saw right away is that there's one system in the cell that is very efficiently repressing our ability to make that G into a C with prime editing, and that's the DNA mismatch repair system. And so these are genes that make up two components that are core to one of the primary mismatch repair pathways in cells. And you can see that when we have loss of function of these genes, we get this big increase in prime editing. And because my lab is a screening lab, I should say we did this screen in collaboration with a fabulous graduate student in David's lab, and then my graduate student, Junyan, did two out of the 4 screens. And so because we're a screening lab, we decided with Peter that we would do this with both the PE2 system and the PE3 system in 2 different cell lines. And you can see across everything we did, whenever we got rid of mismatch repair, we increased the ability of cells to do prime editing. And so at the end of this project, simplifying a whole bunch of work down into one arrow on this diagram, what we showed is that when you have mismatch repair, it actually suppresses this step, the ability to take this edit and put it into the genome. And this gave us like sort of a sort of peek at the mechanism of what must be happening here, which ended up making a lot of sense. So the first step of prime, and this is something that's been shown in biochemical experiments, is that you make this flap with the intended edit, synthesizing 5' to 3'. You have this 3' flap. Our data told us that at some frequency, it must pass through an intermediate that makes a mispair in the genome, something that looks like this. And in particular, the mismatch repair machinery removes the intended edit at the expense of returning to the original sequence and not the other way around, because we infer that there is this nick here in this intermediate structure. And the DNA mismatch repair machinery uses nicks to figure out which is the strand that it wants to fix, right? So the presence of this nick will actually tell the mismatch repair machinery to remove this edit. This is because the predominant place in which mismatch repair works is in DNA replication, and the new strand is the one that is probably, when you have a mispair, the one that probably has something wrong with it, and it's loaded up with nicks. So this is the biological signal for how this works. Of course, in the knockout cells, when we knock out mismatch repair components, we can see this big increase in prime editing efficiency. And then what was interesting is that when we went to this PE3 system, this is this one that has 2 nicks, right? We can infer that the reason that we get this little boost in PE3 is because now the mismatch repair machinery at some frequency, when that second nick is there, doesn't know which strand to fix. And what's neat is that when we get rid of mismatch entirely with these knockouts, we don't see a further increase in PE3 compared to PE2, suggesting that a lot of what we're modulating here is just the activity of mismatch repair itself. So this gave us some biological insight. It's also a fun toy that we're using in the lab now to study mismatch repair itself, because it turns out that this is a fabulous sensor for what the mismatch repair machinery is doing. So when we went back into the mismatch repair literature, we started digging around in the old literature. This is a paper from Paul Modrich's lab, who won the Nobel Prize for his work on mismatch repair maybe 10 years ago now. And what we saw in a lot of his work was that different mispairs are seen by the mismatch repair machinery with different frequencies. And so from this paper, what we see is that CC intermediates are extremely poor substrates for mismatch repair machinery. Okay, and so what that means is when we're trying to make a G to C edit, actually the CC intermediate shouldn't be seen all that well by the mismatch repair machinery. And this bears out in our data. because we can look at all the different edits that we're trying to make and the mismatches that should be in their intermediates. And we can see that relative with and without mismatch repair, these G to C edits, they don't always actually get sensed by the mismatch repair machinery. And we've shown and worked in the lab since in doing these sort of massive screens to look at the way that the mismatch repair sensors pick up these different types of edits, that if we're trying to make a G to something substitution in either mismatch repair proficient or deficient cells, you can see that G to A and G to T actually are really suppressed when you have mismatch repair, but can jump up to really high rates when we get rid of this mismatch repair. But the G to C edits sort of live over here and do equally well with and without this machinery. And what that tells us is that the mismatch repair machinery just isn't seeing those intermediates as well. And so right now in the lab, we're using this as sort of a sensor to see and study sort of what does the mismatch repair machinery see and not see inside cells. Okay, one of the other things that we did during this project is we figured that actually if we just had a way to suppress mismatch repair when we do prime editing, that would make it better. And so Peter and David's group engineered this dominant negative MLH1 protein that we could co-express with the prime editing machinery, making, I told you the nomenclature is kind of crazy, making the PE4 and the PE5 systems respectively. And what we saw is that indeed, when we express this transient suppressor of DNA mismatch repair, yes, we can get these rates to increase across many different edit types and different cell types. June in my lab at the time was doing these experiments. We were reasonably excited about this because you can see the background of prime editing HeLa cells, K562s and U2Ss was extremely low. This is about 10% editing. So a lot of these are like 2% editing in these cells. And we could actually get measurable, meaningful increases. But June at the time was looking at these rates and thinking this is still not great, right? We don't love only getting 10% editing in a lot of these cells. And so he was thinking at the time, surely there must be something else we can do here. There's other reasons why inhibiting mismatch repair is not sort of the ideal strategy from a biotechnology standpoint to improve things. And so June set out to do a set of genome scale screens. In the first set of screens, we were only looking at the influence of DNA repair factors on the system. And so June said there must be something else in the genome that is either mediating or inhibiting our ability to do this type of editing. And so what June did is he built out a reporter with a GFP that is off, and it's off because there's no start codon in these cells. He put this reporter into K562 cells and showed that when you actually prime edit in a start codon, you can get some frequency of them to turn green because now they're expressing the GFP. We can see this in our flow cytometry plots. So in gray here, this is a histogram of cells that are unedited. There's no green cells in this population. But when we do our editing, we get this very nice set of cells that express GFP, and we can increase the frequency of those by getting rid of the non-suppressor MSH2, which is part of the mismatch repair machinery. The other nice thing is as soon as you have a flow cytometry assay for something, we can think about doing screens to validate that actually he could sort out the cells that are GFP plus from the ones that are GFP minus, and that this reflected the ability to prime edit. He did that sort, he pulled out the GFP plus cells over here, and in green, what we're representing is the intended edit in those cells, and you can see this huge enrichment of the proper start codon edit in the green cells. And then when we look in the GFP minus cells, we see that there's no editing. And so,
ask a dumb question.
Yeah.
Did this new approach, like the larger?
I don't think I've ever heard you ask a dumb question, so I'm waiting for it now.
Did this require a selection-based screen at the phenotype level, as opposed to what you did previously with just sequencing?
Yeah. So here we're
guide and the.
Yeah. So before we were doing the sequencing, right, which, to be honest, in a lot of systems where you get a whole bunch of other edits, gives you a lot of information, but didn't give us much information in the prime editing space.
There's a lot of sequencing information on that stuff, whereas here you're just enriching.
Yeah, and so
edits.
That's right.
And that was needed because of the size of the library here.
There's pluses and minuses to both. So actually, we have much higher sensitivity in the sequencing-based assay. So anything that gives us, we've actually gone back and done the comparisons. Anything that gives us a small fold change, we can pick up with repair-seq really, really well that gets lost in the noise on these assays. But anything that is, we can't, in the repair-seq assays, we don't get rid of dead cells. So it turns out that anything that is killing your cells and impacting your ability to edit also sneaks through the screen. Whereas here, because it's a sort-based assay, we first select the things that are alive, and then we select the things that are edited versus not edited. And in a retrospective analysis, it's actually cleaned up the data set quite a bit. And because, so, and it's cheaper, right? So we have a larger library. But the other thing is that we only in this project cared about big changes,
Yeah.
right? So the sensitivity we just didn't care about as much anymore. But it is, there's a lot of it, because we've done so many of these different ways now, there's a lot of information for comparing technologies and sort of what they're good at and what they're not good at. We've thought about this a lot. Okay, so once you have, as Sam said, once you have a selection like this, you can do a screen. So we're going to go back to the CRISPR-I system. So now we're going to do a loss of function individually for every gene in the genome. It's really about 19,000, not quite all of them. Okay, so we put them in the cells, we do our editing, some turn green, some don't. We gen fact sorted them, and then we were looking for our guides that are enriched in one population versus the other. Okay, and that would tell us whether or not you're promoting or suppressing prime editing. This is what the data looks like. It's an incredibly clean screen. We got a few things that when we knock them down, we improved prime editing, and one that did the opposite. Yeah.
Did you sequence verify that it was like the precise edit upstream of the start codon 2? Like,
Yeah.
Is there any bystander? Okay.
Yeah, so there are bystanders. So that's this data, and we did it for the screen too. So the green is the enrichment of the intended edit. When we do it with PE3, which is what we did in the screen, you can actually see that there's intended edits and indels in that population. It's both. We set the screen up that most indels will turn GFP on when the intended edit is there. This was a design choice because we were actually looking for things that would stabilize the editing components, which this adds to that signal. But if you're looking for specificity, you can just flip the thing around and you can do it the other way. Good questions. Okay, so Sam, your question. If we did a repair-seq screen, we would expect there to be a whole bunch more stuff down here, just because we would expect there to be a correlation between stuff that's dying with stuff that's not edited, right? And so by sorting that stuff out, we actually got a very clean data set. And one of the things that popped out at us right away is that there's this gene. The gene is called SSB. The protein it codes is LA. We're going to refer to it as LA throughout the rest of the talk. When this gene was knocked down, we killed the ability to do prime editing in these cells very robustly. What was neat about this screen is not only was there one clear thing to look at, but when we went to go ask what it is, it gave us a sense that this was something that would be important. So what the LA protein is it is an RNA binding protein that does a large number of things, but its most characterized activity is that it binds the poly-U 3' termini that are left on the end of Pol III transcripts. It is a chaperone for them, but it also protects them from 3' exonuclease activity. Now, if you're looking at something that looks like this, and then you're looking at the prime editing system, and then you also have the piece of information that in our screen we expressed the PEG RNA, the guide RNA here, as is common in the field from a Pol III promoter, you can start to put together what we started thinking about this gene, because prime editing is very dependent on the 3' end of this Pol III transcript, right? In order to get this reverse transcription to go, the template has to be there, which is on the 3' end of the Pol III transcript that should have these poly-U's, and then by definition should be binding to this LA protein.
So as I'm sure you're aware, right, people, put these like 3' secondary structures on the end of PEG RNAs now to kind of try and get ahead of this. Was that true in your screen as well, or?
No, so this was before that, and we'll get to that in a minute, right? So not only have folks put these 3' structures on the end to try to get ahead of this, there's also been some efforts to put like an MS2 loop on the end and then recruit a binding protein to it. We'll show you why we think this matters sort of more than those other efforts towards the end of the talk, but that will come into play. But in this case, no, there is no structure on the end. It's just it terminates at the poly. Okay, so we wanted to figure out what was going on here, but at the same time, we also needed to make sure that the phenotypes were robust. So June repeated the screen conditions with PE3 and knockdown of LA and saw, yes, there's this nice reproducible decrease in prime editing of the intended edit when we knock down LA. This is also true across the PE4 system, the PE5 system, and when we put a very different type of prime edit insertion in here. So this is an insertion as opposed to a 2' substitution. And so this phenotype was really robust across all the different systems and edits that we threw at it. In this experiment, this happened to be an experiment that we did in a site that we knew there was a lot of sort of edits and indels that went into the same target site. We could also see that the phenotype showed up for these type of events, but didn't show up for indels alone, which also happens at this site. And so what this told us, again, it's starting to point to this idea that this has something to do with the RT and the templating, as opposed to the ability of Cas9 to just get there and cut and do something. Okay, so at this point, what do we know about this? We know that the phenotype holds across lots of different systems. We also know, although I didn't show you or mention this directly, that the phenotype is orthogonal to DNA mismatch repair. The two phenotypes don't have anything to do with each other. And what I just showed you is that the phenotype is not apparent in other editing outcomes that also require Cas9. So to sort of further get at that last point, what Joan did, he made a LA knockout cell line. LA is an important protein, but it turns out that you can make knockouts and they grow completely fine in this cell line. And he stably expressed the prime editing protein in these cells. And then what he did is he took a whole bunch of different Cas9 editing approaches and asked what happens with and without LA. So in this plot, what we're looking at is the ability to do editing in an LA knockout cell line relative to a parental cell line, which expresses LA. And we're looking at the relative ability to do editing with an adenine base editing system, a cytosine base editing system, Cas9 double strand breaks, and prime editing. And although we have some effects on other systems at some sites, they're completely inconsistent across the sites that we've looked at, and at several sites have no effect, whereas the prime editing phenotype always shows up. And so what this is again beginning to tell us is one thing that's unique about prime editing is the dependence on this 3' end. We could also do this experiment with just a regular guide RNA that didn't have a 3' end. We can't look at prime editing with this, but what we can see is when we do regular editing with and without LA, there's really not much of a phenotype, right? And so at this point, it's really beginning to sound like the interaction with this 3' end on the PEG RNA is meaningful to the system. So as the Zoom member just pointed out, while we were working on this, it came to our attention that David Liu's group had published this paper where actually they had done some beautiful work showing that if you put a structured motif on the 3' end of the PEG RNA, making what folks in the field call an ePEG RNA, you can actually boost your prime editing rates. And the sort of the mechanism that is thought that this is thought to go through is that there's some sort of stabilization of the PEG RNA in the 3' end here. And so we thought we better test this with ePEGs too. And what we could see is that with the ePEGs, the extended PEG RNAs or the regular ones, we always had this phenotype, right? But with the ePEGs, the phenotype was buffered, and this was especially apparent in the system, not the knockout cells, but in the knockdown cells. So here, either using CRISPR-I, where we knock down LA, we could see that the drastic phenotype we saw with the PEG RNA was really, it was still there, but buffered with this ePEG. And with RNAi, we saw the same thing. Okay, and so what this tells us is that the effect of prime editing is to some extent buffered when the structural motifs are appended to the 3' ends, but in the complete absence of LA, we still see these strong, robust phenotypes. In data I didn't show you is that this seemed to be dose dependent. The more we knocked down LA, the worse phenotype we saw. And then Jim did some really nice structure function analysis to show that all of these phenotypes were dependent on the poly binding domain in LA. Okay, so now I've told you a lot of information about this phenotype. I have not yet told you why you should care about any of this information.
If
I
you
promise
feel
you
very
we're getting
stressed,
there.
if
if you overexpress LA, do you stimulate prime editing?
Yes, because it's dose dependent. So you're getting to why we should care about this, right? Okay, but you also should notice by now that this is all in the context of Pol3 expressed pegRNAs, right? So only we think when you have these polyUs there. So one, we wanted to test whether or not it was dependent on these polyUs, but also a lot of applications of prime editing would use a synthetic pegRNA, where you can make modifications to stabilize the end, right? And where they don't have these polyUs. So in order to get a real deep look at whether or not these polyUs were important, we could actually we got a lot of information out of this structural study that was published in 2006 in Molecular Cell, where the structure of LA bound to a the polyU at the end of just an RNA oligo was solved. And so we knew the residues that were interacting with this, the U's at the end of these Pol3 transcripts. And then it was also shown that if you put something bulky specifically on the 2' hydroxyl group of the terminal uridine, you actually lost the ability, the LA bound with much lower affinity to the ends. And so just to test to make sure that we think that this is the interaction that is relevant here, what June did is he designed synthetic pegRNAs that had different modifications with or without a 2' O-methyl group, this sort of bulkier group on the terminal uridine. And sure enough, what we saw is that if you were a pegRNA that had stabilizing mutations at the end, so these are the standard stabilizing mutations for synthetic pegs in the field. These are several 2' O-methyls and phosphodioate bonds. If you looked like this, you had no U and you had this bulky 2' O-methyl group on the end, whether LA was around or not, you got the same amount of editing. This cell didn't care. But the minute you put a free 3' or a 3' uridine with that 2' hydroxyl in an arrangement where polyU could bind, that's when the phenotype came back. And so we think this gave us good evidence that in fact it was this physical interaction that is mediating the phenotype here. And we saw this both at this one site and across all the different sites that we tested. And in fact, when we tested U's with and without the ability to bind, we saw this big difference between the occurrence of the phenotype and not. And so this is starting to robustly tell us that this interaction is really important. But again, at this point, you should be thinking, so what? Who cares, right? And this is where I have to give a little bit of a shout out to the editors at Nature, because we sent them this paper. We thought it was really interesting. And they sent back an email that said, so what? And so we went to the lab and we did the next experiments and we sent it back to them. And then they said, aha, now I understand why this was important in the first place. And so I give them credit for sticking with us on this. Okay. So this is the prime editing protein that everybody uses in the field. This is the Cas9 with the fused RT. And what Jen did is he thought, just as Alberto thought, if it's dose dependent, if we can wreck prime editing in a dose dependent way by dialing down the amount of this protein, maybe by increasing the amount of protein, we can dial up the rates of prime editing. And the easiest way to do this at the time was to take LA, or in fact, the LA binding domain, just that domain region, and just fuse it onto the editor. And so when he did this, this was a really fun day in the lab. What we saw is this is this sort of low amount of editing that we see in most cell types with prime editing. Here is about 5%. And with every single fusion that he fused this thing to, we got these rather large jumps in our ability to do prime editing with the intended edit. And then down here, these are these unintended edits. So they remain very low, but so it remains very precise, but the efficiency goes way, way up. Okay. And then just because this one gave us the most amount in our earlier tests, we started to characterize this guy, which is a C-terminal fusion that we ended up calling PE7. You'll notice that the nomenclature skips PE6, and that's because that was published a little while ago and has nothing to do with us, so it's not in effect. Okay, so we can look across, so these are in U2S cells, and you can see with the standard prime editing situation here with PE max and our pegRNAs, we get very low rates of editing in these cells across all these different sites. But then when we use those EPEGs, this is the improvement from EPEGs. So these bars here, they're very sort of meaningful but low improvements. And by contrast, when we put PE7 in with our PEG RNA or our EPEG, sorry, the E is missing here, this should be EPEG here, we see these very robust increases in prime editing across all the sites that we tested. And what is interesting to us, although we don't fully understand it at this point, is that we rarely, if ever, see a combined improvement from EPEGs and PE7. What is far more common is that the best editing is with PE7 and the regular unextended PEGs, because at several sites, what we see is that the use of the EPEG with PE7 actually compromises editing. We think this is something to do with the structure impeding the ability of the LA protein to actually bind onto the end. I'm not quite sure why this is, but it seems to be robust across all of our data. Okay, we then tried this with all the different prime editing approaches that existed at the time in the field. And what we can see is that this is the improvement with PE2. It's robust, but it's pretty minimal. We can see as we go to these more efficient prime editing systems, we can get this really nice, robust improvement between PE max and PE7. And to put this in context, the first study I showed you with mismatch repair, this was the improvement we were really excited about. So we're using PE max here, and we can see the difference between editing with PE max and in the presence or absence of mismatch repair was the difference here. And to put that in context, this is the biggest sort of maxed out improvement we see with the current technology. And so this is pretty fun. All right, just to beat a dead horse, we figured why not go back with PE max and make sure we feel like the interaction is the thing that's going on here, and this is what's mediating the effect. So we dug back into the structure. We mutated 4 of the residues in the LA binding domain that actually make the residues that interact with the poly-U. We mutated these to something that should disrupt those interactions, and we see that this big increase we see from PE max to PE7 completely disappears when we get rid of the ability of that LA binding domain to bind the 3' poly-U. So at this point, we think that this is really the key to what's going on here. So a lot of tech dev, but we then went on to do all the things that you have to do to convince someone that this is a useful tool. We showed that when you use PE7, you edit with PE7 relative to the state of the art in the field. There's a negligible effect on cell viability and growth, that we really don't have many changes in the transcriptome, and that we increase off targeting only at a minority of sites. And so if you're interested in the effects of this, we have a lot of opinions about how max efficiency also relates to your ability to detect an off target. So if you're interested in that, I can probably talk your ear off about something trivial for a while. Okay. At this point, now you should all be thinking that this has all been done in toy cell lines. We've talked about 293Ts, we've talked about K562s, we've talked about U2OSs, but we were really interested in trying to explore whether any of this would be therapeutically relevant for therapeutic applications of prime editing. And so what we did is we turned back to this synthetic guide RNAs that I told you about. And that's because these are ones that are in the therapeutic space. There's a lot of sort of delivery applications, but one of the ones that is sort of important in that space is delivery of synthetic guide RNAs. And so what I showed you before is that when we have this free U, that this configuration on the 3' end that LA can bind to, that we think it does bind to these synthetic guides. And so what we did is we combined the PE7 that's fused to the LA binding domain with what we think are these LA accessible PEG RNAs, the one it can bind to. And then across different sites, we looked at sort of what the relative levels of precise editing would be. And across all the sites we tested, we saw that combining these two things together gave you a meaningful boost in all cases. And in some cases, not depicted here, but up to about an order of magnitude improvement. So those were also in toy cell lines, but then we had a fabulous collaboration with folks in Alex Marson's group at UCSF, where we showed that this trend was very true when we applied this technology to editing in T cells. And you can see that even at some sites, we're getting editing, very specific editing with the system up to about 50% in about 50% of cells. And then with Dan Bauer's group at Boston Children's Hospital, we also tested all these systems in primary hematopoietic stem and progenitor cells. These are these ones that are really relevant to the sickle cell treatments. And although we don't see huge amounts of editing in these cells, we robustly see that using these tools gives us a much better improvement over sort of the current state of the art for these. And I should say that we tested EPEGs in this system as well. Okay, so with that, we've sort of given you, what I've done is given you a tour of all of the prime editing work that we've done in the lab. I think it's really remarkable, actually, how quickly this field has gone from something that worked and was very precise, but worked very poorly, to sort of this steady march towards something that's working much better. The story I told you today is about how LA is a major promoter of prime editing when it can be, when it can by the ends, how we can use an interaction between LA and the prime editing to support and improve prime editing. And also this thing that I just alluded to is this idea that despite remaining challenges with efficiency, prime editing has proven to be very tractable for technical optimization. So a lot of the HDR-based approaches where we were trying for decades to get closer and closer to precise editing, they took a lot of work and a lot of optimization. Whereas this tool, it seems like every time we look at it from a different angle, we have some new idea for how to make it better. And so we're pretty bullish on this technology. To end up here, there's many things you can do with these tools. I'll give you a peek at some of the other things that our lab has done. In collaboration with a developmental biologist at Princeton, who would love to do very precise, very efficient editing in embryos for studying the earliest phases of mammalian development. We've applied the PE4 system, and by suppressing mismatch repair in that machinery, been able to show that we get really remarkable rates of, precise editing in mouse embryos. We've also started using a lot of our optimized prime editing tools to do single nucleotide variant screens in the lab. And so this result is the result of a library of about 250,000 PEG RNAs, where the ones in red are all targeting stop codons to essential genes. And then for every single one of those, we have matched synonymous controls. And you can see that these screening systems are really starting to work pretty well, where the synonymous controls give us very few phenotypes, and a huge number of stop codons in the library are giving us phenotypes. And so we think we can really start to do some variant screens with these systems. And with that, I will thank you for your attention. There are an enormous number of people to thank, primarily June, who stuck with the LA project all the way through, and members of my lab and collaborators, as well as funding sources.